Hello, thank you for the organizers. Hear me well, hopefully. Um, I'm excited to share with you the CRCNS grant on dynamics of thalamocortical network during sensory discrimination. And this is not working for me. Mic should be on. Oh yeah, okay. All better? Okay, yeah. Much better. This is not. Okay, and this is a joint collaboration with our side in the Hebrew University in Jerusalem, the experimental side with Professor Mark Ryan of the Michigan State University, more on the computational side. And what's so interesting about thalamocortical networks? So,
You stand there.
oh, you want to see me also? So, what's so interesting about the thalamocortical networks? Well, I think we know already that thalamus is not just a relay station. It doesn't just transfer information to cortex. It's a loop. It's a thalamocortical loop going back and forth from thalamus. And from our standpoint, there are lots of loops within cortex thalamus. There are lots of areas in thalamus and cortex. And that gives us motivation to look at the thalamocortical network in a wide sense, not just one channel. So to do this, the experimental side, we combine between two imaging techniques. So this is a mouse, you see it at the top here. This is a mouse, and on one side we have light field imaging, we expose the skull, and these are transgenic mouse lines that express calcium, GCAM6 in layer 2-3 excitatory neurons. The whole dorsal cortex is accessible on the left hemisphere, and on the right side, we use that right hemisphere to insert 32 optical fibers, multi-fiber photometry, simultaneously 55-degree angle to target 13 different time nuclei, and since we're on that time, we can also go to a amygdala area. So we have a wide range of thalamocortical networks. You can see here that sketch of the brain. And what's interesting about this, once you have a wide angle, you can look wide, you can see modality-specific areas like paracortex and S2 in whisker, and in thalamus, VPN, and PO, which are whisker-related. You can see auditory areas, A1 in cortex, and MGB and NGB in thalamus, and also for visual. What's interesting also, you can simultaneously look at higher order areas, lower order areas, for example, paracortex and S2, RL for whiskers, VPN and PO in thalamus for whiskers. And what I like, since you have a wide, almost continuous different cortex, you can look at these axes, for example, anterior posterior axis in both cortex and thalamus. So I'm going to talk about two projects. First one is higher sensory processing, watch versus work pathways, and the second project is different sensory modalities, auditory versus tactile. So what and where system? That's classical 40 years back. In the primate and human visual system, we think there is what ventral stream, which encodes in the visual system the object identity, what is this object, and the dorsal stream, which encodes where that object is guiding action. Jump to the mouse system, we want to use a whisker system because mice can actually locate quite well with their whiskers and discriminate quite well. And based on anatomical studies and some of our work, we think that from paracortex to RL, there is a divergence to the frontal pathway. That's kind of the where pathway to M2, and the other pathway to area P, posterior areas. That's
Go ahead.
yeah.
That's
the what pathway. Okay. So, but the involvement of other areas in subcortex, for example, in our case, thalamus, is relatively unknown. So to do this, we trained mice on two different tasks. The head-fixed mice. It's a what task. The mouse needs to use its whiskers to discriminate between two different textures, or a where task. The same mouse is trained on another task where it's the same texture in a different position, both positions touch the whiskers. One is go stimulus, the other no go stimulus. And the mice can do this pretty well, both tasks. And what's interesting is that if you look at one of in this case, the gold stimulus, it has an identical texture and identical position. So you can compare the same stimulus under different contexts, either a what context or a where context. And just to make things a bit more complicated, okay, we think by maybe that this is the what and this is the where, but we actually know already from previous work from our lab that it's not so trivial. That actually what the mouse is actually doing. If it's an active strategy, it's moving, sensing the texture. He can maybe use the frontal area and a passive, waiting for the texture to come to him, still sensing and performing. It can be area P or the posterior passive. So we're asking what's what, where, or active passive. So just to introduce quickly, not all of the PhD student, she's responsible on this part. The mouse is moving during the task. That's very, very, very important for us to understand, especially this wide neural dynamics. So the mouse is moving, it can move during the task. That's an active trial, whisking a lot, moving a lot. But it can also be very passive, not moving much and just moving during the reward only. And it turns out that different mice use different strategies, and some to different extents. Some are just looking how much the mouse moves here in the active, in the sensation period. Some are active, some are passive, some in the middle. What we want to know that at least between what and where task when the mouse is performing, there is not much difference in strategies, insignificant at least. How does the brain look like? So let's look at cortex first. This is just activity maps, activation maps, delta F over F during the 4 cases. What's active versus what passive?
Sorry, just gotta turn your mute.
I was muted until now.
No,
When you start over?
the people online are getting a mute.
But you can see that.
Getting an echo. Yeah, so we want that off, but I can't figure
You
it out.
want to turn it off?
Yeah, because this one's picking it up. Okay, thanks.
So somebody online tell me that's okay, right? Chat. Okay, so the activity maps for what and where, active, passive, and you can see the main differences between active state and passive state, regardless of the task. And just take the difference, so the clicker, once this is, okay. Take the difference activation maps, active minus passive. You can see red, that's active bias, and blue, that's passive bias. This differential map for one mouse, repeating over mice, and just to quantify this, just take the differential response, active minus passive, and arrange cortex from posterior to frontal. You can see that posterior areas are more biased to the passive, and frontal areas are more active in terms of bias. That's cortex, but thalamus, that's kind of more novel. And we see the thalamus act the same. If you look at the gradient from posterior to frontal, within the thalamus, you see posterior thalamus more passive and anterior thalamus more active. That goes one-on-one with cortex. And to look at that as a network, because we're simultaneously measuring these dynamics, we can look at the full correlation matrix between all pairs. We just calculate the correlation between each pair. For the active case, for the passive case, this is a differential correlation, active minus passive. And this is the matrix for cortex, just starting with cortex. And just order that, do some hierarchical clustering, we get 2 clusters, 2 groups. If we superimpose the groups back on cortex, one group is mapped onto the posterior cortex, it's blue, so it's more biased to the passive. The other is kind of frontal, more biased to the active state. So you get this posterior frontal divergence. You can continue that to thalamus, and we can calculate what I like personally is the seed correlation map. Just taking a seed area in thalamus, for example, a frontal one, RT, and just correlate that during sensation with each pixel in the cortex, and these are correlation coefficient values. So RT is more positive according to the frontal cortex. Another area For example, MGB, posterior thalamic area, is correlated with the posterior cortex, also BLA. And just to do the same analysis and extend that to the thalamic areas, you can see thalamic posterior areas are blue with posterior cortex, more passive, and frontal areas in thalamus with frontal cortex, more red, more active. Right. But these are just me telling you active, passive states, but there are much more conditions and parameters that we can look at for the task and for the behavior. So we just calculated a multilinear model that we have the task parameters, behavioral parameters, how much mouse is moving, licking, performance, task, choice, history, all these 14 parameters. And we try to say, can we predict that based on these parameters, the neural dynamics in each cortical or thalamic area? And eventually you get these beta coefficients. Non-zero values mean something, that there is some correlation, positive or negative. And you can also look at the unique R. I'm just quickly going to show you what happens in the beta coefficient. So for activeness, the parameter, the predictor, can predict, have a positive beta coefficient in the frontal cortex, negative in posterior cortex. On the bottom here, you can see what happens in dynamics over time. For thalamus, also this posterior frontal axis in terms of beta coefficients. Performance also more frontal, and in the posterior thalamus, more negative beta coefficients. And history, what happens in the previous trial with the correct projection, for example, so positive values in the posterior cortex, also posterior thalamus, and negative values in frontal cortex, in frontal thalamus a bit. Task parameters also, I'm not going to go into the whole study here, but just telling you that, okay, there is much lower coefficients, much lower unique R's, also R squares, but still we do see some posterior thalamic area, the posterior cortical area that actually reflect or can predict the task. And just, if you want to take the subspace, we have a vector of beta coefficients for each area, and we want to say how similar are they across different thalamic and cortical areas. This is just a reduction of dimensionality in the T-SNP plot, and you can see two clusters of frontal areas, posterior areas, both in thalamus and cortex, and frontal areas in thalamus and cortex. 5 minutes, that's good. Okay, so we find frontal and posterior thalamocortical subnetworks, which encode distinct parameter subspace, not only active and passive strategy. So the second project, again, was the 5 minutes that I have. Just to remind you, we want to look at auditory versus tactile. So again, tactile has its areas, classical areas, and auditory has its different, maybe similar, we'll see in a second, areas. And this is led by Yoav Levy, an MD PhD student in the lab. So Yoav trained another 5 mice on a tactile task, go, no go, as you saw before. One texture is a go, the other texture is no go. And he switched also the textures, the same mouse with auditory pure tones, one go and one no go. The task structure was almost identical, and the behavior did not differ much. The mice can perform the task quite well, both tasks. And also what's important is the movement. I just talked to you about the strategy. So movement is important. We're going to just concentrate for the sake of this talk on what happens for passive trials. And there, when the mouse is not moving much, not significant between two tasks, you can actually compare better, to some extent, auditory and tactile responses. So now we look at the time course. Again, thalamus and cortex, you can see all kinds of dynamics when we compare tactile hits versus auditory hit trials. Again, in the passive case, not moving much. What I do want to highlight is some thalamic areas. So for example, MGB, that's an auditory thalamus, classical, lower order, higher in hit auditory compared to correct projection auditory, and not much in tactile. VPM is kind of the classical, not lower order whisker, higher in hit compared to correct projection in tactile, but not in correct projection. What I like about it, since we are wide, we can look at these higher order thalamic areas, for example, LGD, which is not so whisker, not so visual, higher order, whatever, and it actually encodes both. So it encodes, doesn't matter if it's an auditory or tactile, it encodes higher, significantly higher hit compared to projection. So these higher order areas, maybe we want to think of as a generalization of the task or the choice of the model. To finalize last slide, we can look at these seed correlation maps. Again, take a seed in the thalamus somewhere, this, for example, the LGD, and you can see that in some cases it's very different. Here, LGD is positively correlated with the frontal cortex, negatively correlated with the posterior, and the auditory task is the opposite, more cortex, more correlation with the posterior cortex. So I actually don't know what that means. I'm just showing you that there are differences within the same mouse performing the task in the same thalamic areas. MGM, for example, different pattern of activity. So we can take these maps and say how similar and different are they across tasks and across thalamic seeds or also amygdala seeds. And again, this is just a T-SNP plot reducing the dimensionality. We see mainly 3 clusters is already average across all the 5 mice. And you can see some areas, for example, we think each cluster is frontal, posterior, and more broadly tuned correlations. We see some areas jumping from cluster to cluster, LGD, MGM, for example, but some thalamic areas are stay within the clusters across auditory and tactile. So each cluster has both auditory and tactile seeds. And that's it. So to summarize, we saw that thalamus and cortex are engaged in a wide range of interactions. We saw that thalamus and cortex display anterior-posterior gradients that are mostly, but not only, related to behavioral strategies and dominated by behavioral strategies. And we saw that thalamocortical interactions of different sensory modalities are complex. And for me, it's really the take-home message. We have to look wide, to look at many areas together. That's kind of where I think is the juice. Okay, so thanks to the lab members, collaborators, especially Mark Reimers on this project, this funding for this project and others, and thank you for your attention. Questions?
How do you think
I can. Go ahead. You can.
how do you think that the clustering would change if you had a combined task where there was tactile and auditory information provided at the same time?
So what would the mouse need to do? You would need to kind of
If it was discerning.
attend, or maybe attend. One is a distractor.
Yes.
That's a good idea. I don't know. I actually really don't know what you would find. But I would presume that maybe these high order thalamic areas would cluster together, for example, because they don't care about the modality. But the distractors add on all kinds of stuff.
You can start sharing your screen if you want.
Yeah.
So we try to look at the correlations between these regions and see how they develop in the tasks.
So correlation between pairs. So I showed you some correlation between pairs of regions. That's what you saw, and how they develop.
Yeah, which regions are more correlated with which, and how does that relate to? You were looking more at activity of co-activity.
Yeah. So on the correlation network, I did show you some regions. I mean, cortex is more highly correlated. We have to kind of normalize that, and you see some correlations between thalamus and cortex, which are highlighted in certain tasks and not in other tasks.
Okay, great.
Thank you.